Assalamualaikum and hello. My name is Nurul Husna binti Zainuddin and my Matric number is 153420. I am a second year student of Pure Biology and this video is for BOI207 General Microbiology, and today I will demonstrate on how to transfer bacterial culture from mixed culture broth to an agar plate while practicing the correct aseptic and streaking technique. According to Healthline, aseptic technique is using practices and procedures to prevent contamination from any unwanted pathogens. It is a very crucial in this experiment, but in real life, this technique is essential especially for the doctors to perform surgery so that the patient will not be contaminated. But before starting the experiment, please ensure to always wear a lab coat and a pair of gloves when handling bacterial culture or any chemical substances. But in this video, I'm not wearing any gloves because I don't have it, and let us just imagine that I'm actually wearing one right now. But before starting the experiment, let us see what do we need for this experiment. So let us see what's the procedures of this experiment. Firstly, ensure that our lab bench is sterilized or sanitized by wiping the surface with a disinfectant and a clean tissue or cloth provided. It will remove dust or any unwanted bacteria on our lab bench. So this is the disinfectant spray, and then let us wipe our lab bench so that it will kill any unwanted pathogens or bacteria and also remove dust from our lab bench. Just wipe it and make sure you don't miss any space. After that, don't forget to throw the used tissue in the dustbin provided so that our lab will be always clean. Next, to create a sterile environment, a Bunsen burner is lit up. But in this video, I use a candle as a substitute of Bunsen burner. But basically, to light the Bunsen burner, use the spark lighter to light up the flame and adjust the color to get blue flame. For your information, yellow flame is the coldest, orange flame is medium, while blue flame is the hottest. When the burner is lit, it will create a sterile air. Next, you might need it to use the inoculation loop, but we have to prepare our plate first. To prepare the plate, so this is my plate, as you can see here. To prepare the plate, I will label the bottom of the plate with abbreviation NA, which stands for the type of medium that we are using. NA stands for nutrient agar, and my name, the date of the experiment, and then label the bottom of the agar plate with section A and D. Section A to D, which is the section that we will streak later. So as you can see here, I already labeled the section from A, B, C, and also D with a permanent marker so it won't be wiped out from the agar plate. And then it's time for us to use our inoculation loop. Take the inoculation loop and flame it. Please ensure the loop to glow red because when it glows bright red, due to the high temperature, it will create any pathogens that live on the loop. And after we flame it, let the loop cool. If the loop is not cooled yet, it will sizzle when you dip it in the meat's culture broth later, which might kill the microorganism in the culture. Please ensure to not put our inoculation loop everywhere after flaming it to prevent any unwanted contamination. But if you do so, please flame it once again to sterilize it. Next, this is my mixed broth culture, and then ensure that this will not touch the surface of our lab bench. Carefully open the tube and then flip the mouth of the tube lightly, and then carefully insert the inoculation loop to get a drop of culture. And then carefully remove the loop. Flame the mouth of the tube again and replace the cap. And then we can put the culture back in the rack. Next, carefully lift the lid of our agar plate with other hand that is not holding the loop. In my case, I am holding my loop with my left hand, so I am opening the lid of the plate with my right hand. Please do not set the lid down anywhere and try not to breathe directly on the plate while it is open. And then streak the loop on the agar surface in the section A back and forth over the surface of the agar plate. And then flame the loop again. And then use the cold loop and streak on the section B. Carefully rotate it so that you won't set the lid anywhere on the table. Carefully streak at the section B, overlapping with the section A a few times. And then flame the loop again. Let it cool. Rotate our lid and streak at the section C, overlapping with section B back and forth a few times. Flame our loop again. Let it cool, and lastly, streak on the section D, overlapping with section C. Flame the loop before setting it down. And then that's it. You can set it down on your lab bench because we are not using it anymore. The plate that we have streaked will be incubated invertedly at 37 degrees Celsius for at least 1 day before observing the results. Before finishing the experiment, please turn off your Bunsen burner and place it aside to avoid unwanted fire accident. Before finishing this video, let us recap the safety precautions and also the aseptic technique that I have done before. Before ending your experiment, ensure to wipe down the workbench with disinfectant once again to prevent unwanted pathogens contamination. Carefully wipe it and ensure to wipe all of the surface and don't miss any. That's it. Don't forget to throw the tissue in the trash bin provided and dispose your used glove in the specific bin in your lab. Now let's recap the aseptic technique that I had practiced before. Always wear a lab coat and a pair of gloves when doing experiment. And then before inserting your loop in the mixed cultured broth test tube, flame the inoculation loop until it glows bright red because the high temperature will kill any unwanted bacteria, microorganisms, or any pathogens that live on the inoculation loop. And then, before inserting the inoculation loop into our test tube filled with mixed culture broth, we need to flame the mouth of the tube, and then once we get our culture with the inoculation loop, and before replacing the cap once again, we also have to flame the mouth of the tube. Always ensure that we do not set our inoculation loop anywhere on the table after flaming to avoid contamination from the bacteria on the surface of our workbench. But once if you set it down and you have to use it, you need to flame it once again. Next, don't expose our agar plate to the air as it will contaminate it with the microorganism or dust from the air. Once you open your agar plate lid, don't breathe directly onto the agar plate. And when you incubate the agar plate, you have to incubate it invertedly to prevent water droplets that will potentially contaminate our culture. Last but not least, wash your hands with soap at the washing station before exiting the laboratory. Always practice all of these safety measures and also the aseptic technique so that our work will not be contaminated. That's all from me. Thank you.